Good morning, good afternoon, good evening, depending where you're joining from today. So on behalf of Dariada Drug Discovery, I'd like to extend a warm welcome to our expert panel and yourselves, the audience. I'm Adam Davenport, Chief R&D Officer at Dariada, and your host for the webinar today. So before introducing the panel, I'd just like to briefly cover a couple of housekeeping topics. So the first is technology willing. We plan to conduct a 2-question poll on covalency and then share the results at the end. So look out for the 2 pop-ups, please, and they're simple yes or no, that. And then just to quickly advertise, we'll follow today's sessions with a further 2 seminars. The second one on chemoproteomics on the 5th of October, and then one on degradation the 2nd of November. So please look out for the flyers and invitation. So it's hugely exciting to open Dariada's inaugural webinar series, and this first session, exploring recent advances in covalent drug discovery. So I'd like to briefly introduce our esteemed expert panel. So that's comprising Adam Gilbert, who's Executive Director and External Accelerator Lead at Pfizer. So Adam's contributed to the delivery of covalent inhibitors, modulators, and is co-inventor of Lipfolo, which I think we'll hear more about today. So that's the JAK3 inhibitor program for that. We also have Chris Smith, who's Executive Director for Drug Discovery at Mirati Therapeutics, where Chris drives the work on the targeted oncology solutions for the genetic drivers of cancer. And he really has pioneered fragment-based approaches in diverse therapeutic areas, including covalency. We also have Dan Nomura, so Professor of Chemical Biology and Molecular Therapeutics at University of California, Berkeley. So in addition to this role, Dan is co-founder of Frontier Medicines and also Vicinitas Therapeutics, where they've used approaches such as chemoproteomics and artificial intelligence to tackle the undruggable proteome. And as I'm sure this audience is aware, multiple awards have been won for that work. And we also have Simon Bailey, who's head of drug discovery at Plexium, where Simon leads Plexium's efforts to identify novel protein degrader drugs and address critical medical gaps. And during his career, Simon's led teams delivering over 20 clinical candidates. So hopefully, I was aware that we're really privileged to hear from such a set of great professionals who will power this webinar. So by way of introduction to this session, I think it's fair to say that covalent therapeutics have made a huge resurgence in the last decade, enabling researchers to develop highly potent and selective therapies for challenging diseases. Here at Dariada, we've been supporting covalent programs of our clients and partners since the very beginning of the organization. And having worked on covalent programs for years, we're familiar with the specific considerations and assays that are really critical for covalent program success. And many of these concepts will be part of the webinar today, where we'll explore the progress made in drug discovery through innovative use of covalent platforms and new technologies. In terms of the agenda for today, we're going to break it into 3 themes. And these are general covalency, then we'll pivot and talk about, sorry, I'm just going to answer the question as well. Then we'll pivot and talk about non-cysteine nucleophiles, and then we just want to wrap up and have a discussion around company-specific questions from each of the panelists and share those case studies and learnings. So moving into the general covalency theme. So there's historic trepidation about the use of covalent drugs by investors and pharma. However, there's also a huge resurgence of covalent drugs with a large number of biotechs now funded in the space and big pharma exploring many targets through this approach. So not least the KRAS G12C. And kind of the first question to the panel related to that is, what does the panel think have been the main catalyst for excitement and renewed investment in this area? And if I could direct that to Chris, I'm sure you have something to cover and say on this topic. Chris, we can't hear you at the moment. Just check if you're on mute. Okay, I've just received a message from our tech support. I think the other panel members are unable to unmute. So we're just going to take a pause for a minute whilst we can get to grips with the technology. And you can tell this is possibly the first webinar we've used driving our sessions, so kind of bear with us, please. Let me just check, Simon, do you have the ability to unmute?
Looks like it.
Fantastic. I wonder, is this a question that you feel equipped to contribute? I know this is really critical.
I can certainly make a couple of comments. As almost an outsider to perhaps some of the more significant catalysts for the resurgence in interest in covalency. From my own perspective, a lot of that has been driven by the success of the G12C inhibitors. I'm sure when Chris comes on, he'll give us the insider track on that. Because I think that illustrates some of the unique features that can be gained from covalent binding to protein targets, namely being able to drive significant affinity in parts of proteins that were previously considered unsuitable for binding because of the large featureless areas of those proteins. And being able to sort of key in and generate a covalent bond as a driver of affinity was the initial success of those G12C inhibitors, obviously pioneered by Shokat, and then picked up by Amgen, Mirati, and others. I think the reduction to practice of that concept of taking a classically undruggable protein and a protein region and generating medically and presumably commercially successful products based on covalency has really been a standout in the field. And I think those of us who followed that story were excited by the science from day one and have been pleased to see the clinical results. And I think the medchem that was done at a number of companies in that area was extremely elegant and addressed many of the themes I'm sure we'll talk about today in terms of the challenges that come with covalent drug discovery. Perhaps one final comment, perhaps there's an irony in that some of the follow-on programs in KRAS, looking at other G12 mutants, don't necessarily rely on forming irreversible bonds with a cysteine, the G12D and V, perhaps A variants. So showing that there is actually a pocket in that protein that can be accessed via non-covalent binding. But I think undoubtedly the success of those cysteine targeting molecules was a massive area of interest to the field.
Thank you, Simon. Can I just do a quick check? Do we have Chris, Dan, and Adam back with us? Okay, let's move on to the next question. We can always loop back, so I would like to hear from kind of Chris, who's worked significantly within that target class and target in particular. So maybe going on to, so I guess as we know, there's a lot of excitement about the use of covalent degraders targeting E3 ligases for use in PROTACs and molecular glues. So I guess the question is really to see about kind of Simon, as you're the only panelist at the moment. Are you excited by this development in the kind of covalent drug discovery space?
Extremely. Plexi may not be familiar to many folks on the panel, but we are a company focused exclusively on targeted protein degradation. We're a 100-person drug discovery in San Diego, and we were founded partly on some proprietary technology platform, but with a particular focus on targeted protein degradation, addressing some of the either known or expected liabilities that come with PROTACs and time cereblon molecular glues. And so, relevant to covalency, one of the things that we're interested in is being able to use covalent binding to expand the range of E3 ligases that can be incorporated into protein degradation processes. Beyond cereblon, VHL, and a handful of other E3 ligases, many of the proteins in that gene family feature large amorphous binding sites that are not well suited to typical non-covalent binding. So covalency stands out as a very attractive approach for ligating those areas. One particular feature, and I'm sure Dan will talk about this, that sort of brings the unique intersection of degradation and covalency. So if one covalently binds to an E3 ligase, that still renders that modified E3 ligase catalytically active. So one of the big attractions of protein degradation is these molecules operate in a catalytic function, have event-driven pharmacology rather than occupants-driven pharmacology. And binding covalently to E3 ligases is one way to preserve that covalency. And that's a very attractive feature. It means it gives you, at least in principle, the ability to redirect novel E3 ligases to degrade neosubstrates and yet retain all the attractive features that come with retaining that sort of catalytic turnover as well. So we're very interested in that. We've got active programs in that. We haven't disclosed information in the same way that Dan and his group and companies have, but we hope to do so in the near future.
Thank you, Simon. Great to have you back, Dan. Just check that you can. Yep, we can hear you. Perfect. So sorry, just jumped around a little bit in questions. What I kind of would like to pose to you is kind of a question around chemoproteomics. As we know, it's come a long way in the last decade with coverage targets growing, with the instrument sensitivity, innovative chemical probes and methods for detection of the covalent adducts. And clearly, you've been a major player in this space. So it's really to understand what you see as the next advances in the chemoproteomics space, please, Dan.
Yeah, so I think over the last many years, since the pioneering discoveries around covalent chemoproteomics by Ben Krabot and others, the field has really exploded. And I think with advances in chemical probe design and proteomic mass spectrometry-based approaches, we now have footholds across really the vast majority of the human proteome. And using covalent ligand discovery approaches, you can really start to interrogate and elaborate on more selective molecules that can interrogate these kind of interesting, novel ligandable sites across the proteome. I think also what really excites me is use these kinds of technologies to enable, for example, targeted protein degradation by developing new E3 ligase recruiters, or enabling strategies for rational design of molecular glue degraders, or identifying permissive E3 ligases that we use in degradation, but also going beyond degradation to develop new induced proximity-based approaches, or alternative approaches towards drugging proteins. In the degradation space, one area that we're starting to get more and more excited about are kind of monovalent covalent degraders that don't necessarily act through glue or heterobifunctional based mechanisms. And so I think there's a lot of different approaches that one can take to functionally manipulate undruggable proteins using covalent drug discovery approaches. So I think it's a really exciting time to explore the full capacity of these technologies to go after drugging classically impossible targets.
Yeah. Thank you, Dan. I guess on a kind of related question, it's really kind of around the covalent glues. Obviously, again, there, as you mentioned, receiving a lot of attention. So do you see kind of, so for the covalent approach, sort of moving on actually beyond protein, do you see kind of potential that this could be a method for essentially degrading RNAs? So looking at the transcription as well. So I'm thinking of the kind of the ribotac sort of technology.
Maybe I can help answer that. I mean, so we, have, like I said, footholds across, the vast majority of the human proteome, including, proteins that manipulate RNA. And so, in principle, it should be possible to use covalent chemoproteomic strategies to develop recruiters against various RNA manipulating machinery, and then link it to RNA targeting small molecule ligands to be able to functionally manipulate RNAs. So it's certainly possible.
Thanks, Dan. Can I just check to see whether we have Chris or Adam join? I don't see a full view. Okay, I take it as a no. So I guess let's move on to another question. And I think this is possibly more in Chris's sweet spot, but hopefully as a group we can kind of talk around this topic. So kind of the reduced selectivity and perceived promiscuity of covalent drug is often considered an issue in the drug development phase. So I guess Simon and Dan, what's been kind of your experience of making selective covalent compounds? So is it a big hurdle as people perceive? What do you consider to be the selective in the ranking of covalent compounds? So when you categorize it.
I can start here. So in a previous life, I worked on inhibitor programs at Big Pharma, and there, for instance, we were targeting EGFR mutants and using covalency there as others have, and obviously in that type of case, where you are targeting the ATP binding site, the kinase, the issue of selectivity over other kinases becomes critical for that precision oncology approach. So through that experience and some others, my own view on selectivity is it's a property just the same as it would be in a non-covalent program, and there are well-established medchem principles that allow you to address that. And I think also importantly, there are assays available, suite of assays available that are both target specific and non-target specific, that allow you to understand the properties of the molecule. And I think the two things are, what are the relative contributions of binding energy from covalent bond formation and from non-covalent interactions in the protein of interest? And then you can certainly measure or use as a surrogate general measure of electrophilicity and the potential for promiscuity by using things like GSA adducts. And so if I was to sort of summarize how I would sort of think about design principle, I think reducing the electrophilicity of a warhead while maintaining sufficient potency at the target protein is one tactic that people can follow. And if you can do that, then I think as a chemist, you've taken reasonable steps to reduce general promiscuity, and I think that combined with the other principle, which again I think would be no surprise to people, which is to reduce overall human dose size, are the two factors I would point to for ameliorating the risk of toxicity that's a perception that comes with covalent drugs. And I think all of those things are to a large extent within the control of chemists, and they're certainly aided by the availability of well-established assays and plenty of literature precedent. So I don't think anyone should be apprehensive or have trepidation about covalent inhibitor programs these days. I think they're just another type of medchem project.
Yeah, couldn't agree more with that, because from my experience, it really is focusing on the Ki aspect for your protein of interest and really driving your high affinity for that, whilst measuring the KNACT and getting that window that you require. I think that's where the value of sort of structurally enabled programs and using CAD and other techniques really come in. So you can really design in the Ki component as I guess, an integrated design team, and at the same time have the understanding as the improvements come on board with predictions of covalent interactions, and using those kind of Ki, KNACT drivers and the assay data behind it really are the kind of the path forward and the critical aspect for kind of reducing that risk of selectivity and promiscuity.
I just also mentioned that, I mean, chemoproteomics, right, has really arisen as an approach to be able to de-risk these covalent molecules. You can really assess the proteomoid selectivity of your covalent molecules and couple that with medicinal chemistry efforts to not just optimize potency against your target, but also in parallel the proteomoid selectivity, not only in cell culture systems and living cells, but even in vivo in mice, right, where you can dose a mouse with a covalent molecule, take whatever organs out, chase with reactivity-based probes, and really assess target engagement, selectivity across disease site, and also across every other tissue. And so, and also you can use all the kind of arsenal of chemical biology approaches, like making alkyne functionalized probes and, directly turning your molecule into a covalent probe and looking at off-target profiles using chemoproteomic strategies. And so those kinds of things I think are absolutely necessary to help de-risk covalent drugs, and to make sure that there aren't kind of off-target toxicological liabilities. Without doing that, I think you run the risk of running into these kinds of idiosyncratic tox type situations where you have some kind of off-target that you just didn't know about, right?
Yeah, couldn't agree more. And just to add a little bit, I mean, having been in the protein degradation field for the last few years, come to appreciate perhaps even more the power of proteomics and chemoproteomics in understanding the cellular profiles of compounds, and the importance of understanding that across different cell types as well. And I think in normal drug discovery, when we use a reductionist approach and use isolated assays and so on and so forth, that's very helpful in designing molecules. But I think, as Dan pointed out, there's nothing like being able to get that full picture of the cellular response of compounds, and in particular different cell types and different cellular contexts as well.
Yeah. So just looping back, Dan, to kind of the molecular glue space specifically. So obviously related to some of your work, do you think that covalency can assist in improving the predictabilities of making the glues?
I believe so, yeah. So, some of the work that we've done were we've taken various protein targeting ligands and started depending on various electrophilic handles to look for chemical handles that enable you to convert non-degradative molecules into molecular glue degraders has allowed us to identify a variety of kind of electrophilic handles now and potential various E3 ligase pairings that allow for highly permissive degradation across a variety of different types of neosubstrates. And so I think, that's just one example where you can start going after target-centric approaches. I think the other strategy around, maybe taking a covalent E3 ligase recruiter that you identify and then morphing that into molecular glue degraders libraries to do similar things to what folks have done with cereblon and, other E3 ligases is certainly also possible as well towards this increasing effort towards rationally designing these things. Also, I think more and more rationally discovering molecular glue degraders through, for example, target-centric phenotypic screens is becoming a more and more powerful approach, and that can be done with covalent molecules or non-covalent molecules.
Yes, certainly from my understanding, in the non-covalent molecular glues, essentially, if you are going for the diversity approach, so rational screening of libraries, certainly there's an additional challenge in how you set up your assays and how you identify and pick out and pull out the actives from the baseline. So it does kind of provide an additional challenge, which I just wondered whether the covalent approach could help to overcome some of that with different drivers, and then you design in your KI component later on. Yeah.
Can I maybe one comment, because at Plexium we're very interested in exploring that approach, and at least my simplistic cartoon picture of this is that having covalent binding between a small molecule and E3 ligase in a sort of forward-looking search type assay, phenotypic assay, reduces the problem of generating ternary complexes to generating binary complexes and identifying those and what's going on. So I think it helps reduce the number of variables. The approach that we use at Plexium is to use large-scale screening in cellular systems. And when we're looking for molecular glues, we look for, as much as we can, the entire cellular response. So we're developing high-throughput proteomics approaches, but we've already developed a high-throughput RNA-seq approach where we can treat cells individually with tens of thousands of compounds and measure the cellular response of each of those compounds and look for change in the transcriptome, and from that decipher whether those changes are due to protein degradation. Because of the sort of multiplicity of that approach, actually having something fixed by generating covalent binders to E3s helps simplify that problem quite a bit. So I think there's, whether as a therapeutic approach or whether as a probe for unlocking or investigating the biology of E3 ligases, there's tremendous uses for covalency.
Yeah, then general question. So obviously, if you're starting kind of a program, you've got your initial hit compound, you're given protein. What approaches would you recommend to more effectively build in selectivity at the early stage of a program? I don't know, Simon or Dan, whether you're prepared to comment on that.
I mean, you mentioned earlier on, Adam, and perhaps Dan as well, having structural information, in any program is helpful, but I think particularly when you're looking for selectivity, and I think when you're looking for using structural information for driving potency, it's a great idea generator. But when you're looking for potency, it allows you to I think, quite rationally probe areas of the molecule where you can expect to have positive interactions with the protein of interest and negative interactions with those that you don't want to interact with. So I think having structural information is important. Specifically for covalency, I think as we talked about earlier, having a suite of assays that allow you to understand both the Ki and the Kinact of molecules against your target of interest and perhaps the selectivity targets is important, because all of that SAR is rationalizable if you're getting the right kind of data. So I think making sure that you have the right kind of assays, as much structured information you can, and then being thorough in the analysis would be helpful in setting a program up.
Yeah, so part of it is constructing the appropriate screening cascade and screening tree of when to bring those assays in at which phase, and then pivoting around the data so you have the balance of a streamlined cascade, but also sufficiently information rich for those selectivity targets that are essential for you, either based on homology or potentially through the therapeutic area that you're going after and known off targets. Fantastic. We've kind of touched upon the kind of concerns with promiscuity, idiosyncratic toxicity. But I just kind of want to just come back to that question just through kind of your experiences and the organizations that you've worked at. Has that been a significant issue for you, or those organizations?
The concern has been a significant issue. That the actuality is less of an issue. So I think certainly in oncology, where many, but not all, approaches to covalent drugs have taken place. Obviously, because of the nature of the patient population, there's considerably more tolerance for toxicity in profiles, which helps. Oncology compounds just generally don't fail for toxicity reasons in preclinical development. Even in patients, they typically only fail for lack of efficacy. So I don't think there's anything particular about covalent drugs that's going to hinder the early stage development of drugs in oncology. The idiosyncratic tox, I mean, again, the sort of received wisdom is that shows up late in development with large numbers of patients. It's unpredictable and kill a program. Again, I think in oncology, that's just not going to happen, or unlikely to happen, just because of the nature of the patient population and their kind of overall health status and what happens to those patients anyway. So at least I think in that therapeutic area, the idiosyncratic tox concern is just less. And as we talked about before, I think in general, driving to low dose drugs is always a good thing. I don't think there's any downside to having the lowest possible human efficacious dose. So I think if you can do that, even in other therapeutic areas, you've reduced as much as you can the risk of idiosyncratic tox showing up in late stage development.
Absolutely, reducing that burden and also your cost of goods at the same time. It's a true win-win. And any thoughts you want to share on that topic?
Yeah, I would say things like idiosyncratic tox are kind of a fancy name for you don't know what your off targets are, so you run into toxicity. And again, I think a lot of that stuff can be heavily de-risked using covalent chemoproteomic strategies. And if there are concerns about, for example, prolonged dosing leading to accumulation of off targets, you can also test that too using chemoproteomics, right? Just increasing dosing regimens and timing of treatments and stuff. So, I think all of those things can be de-risked very, very early, even in the kind of medicinal chemistry pipeline. You don't need to wait until you have something close to a developmental candidate. You can really be optimizing all the different parameters from potency, selectivity, PK properties, all of that in parallel using all these kind of modern chemical biology approaches.
Fantastic. So I think we move on to theme 2. Just to give people a quick update, the kind of tech support are working to rejoin both Chris and Adam as hosts so they can kind of unmute their microphones, and we'll loop back to a couple of the questions at that point. But theme 2 is looking at non-cysteine nucleophiles around that. So kind of the first question to Simon and Dan is there's obviously a huge interest in targeting nitrogen oxygen nucleophiles in medchem. So what promise do you think this approach holds for accessing novel chemical space on targets not previously tractable to covalency? I don't know who'd like to start off on that one.
Maybe I can comment on it. So, there's certainly been tremendous amounts of kind of chemistry efforts devoted to going after, amino acids beyond cysteines. And I think probably lysines are the most advanced in that sense. There's a lot of other types of, reactions that have been discovered for going after, for example, tyrosines or methionines or histidines or tryptophan or other amino acids as well. But I would say the main limiting factor, at least in my view, has been whether these reactive warheads are going to be metabolically stable and compatible for oral bioavailability. Even within cysteines, for example, like we know that things like acrylamides and nitriles are compatible for oral bioavailability. There are drugs that are already in the clinic that have these warheads. But for example, chlorosetamides, which we still have in our libraries because they work fine in cells and they still can have decent selectivity in cells, are very metabolically unstable and likely not compatible with oral bioavailability. And when you get outside of cysteines, that space gets a lot murkier. Even within lysines, it's not super clear which lysine reactive warheads are really going to be the best in terms of metabolic stability, safety, PDPK properties. Once you get out of lysines, it gets even murkier in terms of what kinds of warheads are really going to be compatible with that. So all of those things need to be considered, right? It shouldn't, I think the bar of like whether it can be used as a probe for chemoproteomic profiling and whether you can elaborate on that electrophile to gain selectivity for specific proteins and cells, I think that is definitely achievable now, right? To be able to access a lot of these kind of undruggable targets, and many people in the chemical biology field have really been working on this and really leading pioneering efforts on this. But to go from that to a clinical candidate is then another huge leap. And if your warhead is just inherently incompatible for metabolic stability in vivo, then no matter how much med chem you do on the rest of the molecule, the warhead itself is the main liability, right? So I think that part needs to be further progressed in the field.
Yeah. Simon, anything you want to add to that?
Only echo what Dan said, but less eloquently. I think the inherent problem is all of those other bionucleophiles are less reactive than cysteine, so presumably electrophile is going to have to be hotter, and then that just makes that a balancing act with other properties much more challenging. I would also agree. I think perhaps biased because of the protein degradation interest, that lysine would be the next bionucleophile. I think as a field we perhaps should focus on beyond cysteine containing proteins.
Yeah. So do you see kind of over the next, say, 10 years, do you see kind of a shift of focus, so looking kind of beyond lysine, or from the kind of the concerns over metabolic stability translating into poor bioavailability, that actually it's still in the infancy as a field and it may take longer to transition to start to look at those alternative nucleophiles?
I mean, I think the chemistry will advance, right? Now that there's such a huge resurgence in covalent drug discovery now, and a lot of people are now devoting efforts towards developing new chemistry beyond things that can just be used in bioconjugation reactions. I think, a lot of these things will be discovered. I mean, our lab is working on, many other labs are working on developing new warhead chemistry, right? So I think this is a surmountable issue with enough attention placed on it. Having said that, there's still a tremendous number of cysteines that have been unexploited for drug discovery, for a number of reasons. So for example, we have a heavy focus these days on drugging transcription factors and potentially degrading them using covalent strategies by targeting intrinsically disordered regions within proteins. And historically, those, even if you're able to target these things, they've been shied away from because you're not going to be structurally enabled for medicinal chemistry approaches and such. So I think there's still a huge amount of untapped space, even within just cysteines itself, and certainly lysines, without having to go kind of beyond that. But the more amino acids we can tackle, the more ligandable sites we'll identify, and the more targets we'll be able to pursue.
That's Dan's just made a great point. I think there's so much untapped chemical and biological space that can be had by targeting cysteines that we're just not addressing right now. I think there's plenty of opportunity, and going after proteins which are intrinsically disordered and using covalent binding as a way to generate transient binding sites, or otherwise, I think is a fantastic example of that.
Yeah, certainly opens the door for those kind of allosteric transient binding sites where you can escape some of the kind of reported selectivity challenges going after the orthosteric. So yeah, it's a hugely interesting area of using those probes in that sense. I'm just kind of switching kind of a focus slightly, because obviously we know there's more excitement now about irreversible covalent inhibitors as compared to just a few years ago, 5 years ago. So where previously reversible covalent was seen as the way to circumvent the historic fears of promiscuities, the permanent modification. So what's your reason and what are your thoughts around that excitement of the kind of the irreversible covalent inhibitors? And do you see a renaissance of that?
I mean, I guess, the covalent reversible inhibitors were, I think, very useful in terms of, I guess helping to de-risk some of the concerns that people had with, irreversible compounds. But, since that time, there's so many irreversible, for example, kinase inhibitors and Paxlovid and all these drugs that act irreversibly that are now in the clinic. And again, with the advances in chemical biology approaches like chemoproteomics, you can largely mitigate a lot of these kinds of concerns that people have had around covalent drugs. So I just find, much less concern for having to kind of reverse engineer your irreversible molecule back into a covalent reversible drug. Having said that, I think there is advantages. For example, if you're trying to make a PROTAC and you have a covalent protein targeting ligand, you do lose the catalytic degradation activity if the covalency is on the target you're trying to degrade. And so if you're able to reverse engineer that back into a covalent reversible molecule, then you can pick back up the catalytic degradation activity while still exploiting for example, cysteine reactivity against the target. So again, those are things that people played around with in the field as well.
Yeah, I mean, at least from the initial wave of reversible covalents that went into the clinic did not deliver the promise of greater toleration with efficacy. So I think that's still an unproven concept. I think it's a very elegant concept, but still unproven. And at least, again, from my simplistic point of view, it's just another tool in the toolbox when thinking about the types of warheads that you can employ and investigate. Why not include ones that are reversible as well as ones that are sort of classically irreversible, although I'd argue that they're all on a scale anyway.
Thank you. So switching focus to kind of a third theme, so really talking about your organizations and companies. So Simon, so congrats on all the recent successes in Plexium. Kind of the question is, where do you see the covalent degraders in the drug landscape? So do you think this is a field still in its ascendancy?
I think and I hope the degradation is a field in its ascendancy, yes. Because protein degradation is a clinically validated approach. I mean, Celgene BMS's CELMoD suite of drugs have been successfully treating patients for years, and PROTACs and SERDs, for instance, have been finding clinical success. So I think it's still its adolescents perhaps, but I think it is a clinically validated approach. I think when it comes to the non-cereblon molecular glues, most of the targets that have been investigated, certainly clinically, have been well-established targets. A lot of nuclear receptors, estrogen, androgen, for instance, limited number of kinases. So I think there's still this vast untapped potential of going after drugs which can't be classically approached by inhibitors, that could be targeted by degradation. And in that, I think covalency is like perhaps one of the single most useful tools that we have for all the reasons that we've kind of discussed. What you need in degradation as opposed to inhibition is a binding event, not necessarily a functional inhibition of the function of that protein. You need to be able to bind something in order to be able to get it ubiquitinated and transported to the 26S proteasome for degradation. So therefore, you don't need to necessarily bind into an active site of a protein. You don't necessarily need to have high affinity binding, but what you do need is specific binding for a sufficiently long period to allow the formation of a ternary complex with an E3 ligase ubiquitin transfer and so on and so forth. So I think, if anything, degradation is a field where covalency, is going to be more useful than perhaps sort of traditional inhibitor drugs. So I'm super excited about that.
Thanks, Simon. And Dan, firstly, congrats on the financing of Vicinitas and your many academic pursuits. What's your read on investors' current interest in covalence as a big component rather chemoproteomics and the tractability of targets to covalency? Have you seen a shift in their attitudes?
Yeah, I mean, certainly compared to when I, was first studying frontier medicines, many years ago, the attitude towards covalent drug discovery has certainly, really, really changed. I think, a lot of people, 5-6 years ago even were still largely kind of shying away from, reactivity first driven covalent drug discovery programs. But I think, thanks to the approvals of KRAS G12C drugs and such, a lot of that fear has been allayed. And so, yeah, I think these days, most folks really, are not so trepidatious about covalent drug discovery, especially for things like cancer, where, the treatment periods are going to be acute. I do see still some concern around like chronic, multi-year therapies or things like that, and taking covalent drugs and accumulation of potential off targets and such. I would say the investor worry is more just the current biotech climate, and pure platform plays are not necessarily getting as easily funded these days, as opposed to Nuco that has more advanced assets, and so that has nothing to do with covalent drug discovery.
Yeah, totally. Yes. It's a risk-off funding environment right now, and that's assets, not platforms and approaches. So I don't think, I think that dominates in investor thinking, not the science, unfortunately.
Yeah.
Yes, I think there's a shift from the VCs to invest more in later stage assets rather than the early assets, as well as the platforms that you mentioned. Certainly, we've noticed that shift over the last couple of years, and hopefully 2024 will bring that reversion. So just say a few closing remarks, because we're down into our last 10 minutes. So thank you, Simon and Dan, for your insightful responses, sharing your personal views on these topics, helpful, and for helping to carry the conversation with obviously missing two of our planned panelists for that. So we've heard that the understanding the importance of safety and selectivity in covalent drugs, and how modern research techniques address the potential of target effects, is a critical aspect of covalent drug research. We've also been reminded or been informed of the power of chemoproteomics to really or better understand your off-target profile. And that's really linking into the idiosyncratic toxicity, as we mentioned, that you more frequently identify when you go to large patient populations. So it's one of those kind of death knells for certain compounds in phase 3 clinical studies. We've also talked a little bit about future prospects. So we've heard obviously the outlooks from Dan and Simon on the future of covalent platforms in drug discovery, the potential in treating a wide range of diseases. And we just touched upon moving some of the focus beyond purely oncology from that. Just going to have a quick look at the polls to see the scores and the doors. Yep, so possibly not that surprising given the audience attending and this webinar session, but there's a clear majority of people, their organizations are currently working in covalency, and it's about 30 that aren't versus 99 that are. And of those, there's actually a slightly different split to the one I was thinking of, that there's actually more organizations working on therapeutic areas other than oncology, which is a little bit of a surprising. So they're starting to see that shift in the industry. So just before I wrap up, just a quick reminder that the next webinar, really, we will have the technology platform sorted out by that point. But the next webinar on chemoproteomics is October the 5th and degradation November the 2nd. And then it just leaves me to say a huge thank you, Dan and Simon, and also for Adam and Chris, and to thank you, the audience. So that brings the webinar to a close, and hopefully we'll look forward to seeing or discussing with many of you at the next session. So thank you, everyone.